to go into here structurally, at least for now. There are a number of vestigial structures that are seen in a number of vertebrates that suggest common ancestry with another group of organisms or another group of species, and are often leftovers or remnants of development, for example. And so these are great examples here. This is actually one of the ancestors to what are called cetaceans, or the whales and dolphins. And you can see it's extinct. It was lived around 40 to 50 million years ago, and you can see it had a beautiful pelvis and typical limbs of something that's kind of at least amphibious or partly terrestrial, right? And obviously over that 50 million years, these particular groups of animals became more and more aquatic and obviously marine. And now the only thing left of the pelvis is a really small group of bones in the same place because they are pretty useless, right? But they do develop, they just underdeveloped because there are genes that have to turn on, lay that bone down in order for other genes to jump in and finish development of that part of the body axis. So it's a great example, these vestigial structures, of common ancestry, something we're going to start talking about here now with phylogenetics and taxonomy. Everybody's favorite part of 155 was phylogeny, right? Nobody likes phylogeny. I like phylogeny. Actually, I'm not a huge fan of phylogeny. What's really funny is that I was the same way. I'm like, oh gosh, phylogeny is so tedious. And there are biologists that love it. But of course, I've now published papers that include phylogeny. So, you know, it'll come back to you, particularly if you stay in biology. We're going to talk a little bit about the differences between classification, like what they call taxonomy, and then what phylogeny is, and kind of how it's easy to mix them up because as a biological community, we're trying to link the two. And sometimes it's pretty easy to do, and other times it's almost impossible to do because of something we talked about yesterday, which is convergence, right? A lot of organisms look alike, but don't share a common ancestor. And that's really the difference between the two, is classification groups animals based on appearance, what they look like. It doesn't care about anything else. Oh, these two fish look alike. We'll put them in the same taxonomic group. Phylogeny cares only about evolutionary history, kinship, family membership in the process of natural selection. So you got to be able to trace the organisms back to a common ancestor. The problem is two organisms can look very much alike and not share a common ancestor, or two organisms can look really different and share a recent common ancestor because the environment can be quite influential, right, on what an animal looks like we talked about yesterday. So we're going to kind of dive into these and start from the beginning and then work our way down because they're both very useful, particularly when we're doing this stuff comparatively. You know what I mean? That's why I'm kind of going to spend a little time on it, because we're going to be comparing vertebrates, and it makes a lot of sense if you kind of understand, you know, kind of how they all are related to each other. So this guy invented classification back in the day. He lived in the 1700s, very religious guy, an aristocrat, and you know, didn't believe in evolution. He believed in what the church taught him, that all animals are perfect. They were all put on the planet at the same time and so forth, kind of like we talked about the creationist view. But he did start to group animals, and the way he grouped them is still alive today. That is, we still name organisms the same way. So if you were to go out and find a new frog in an Indonesian rainforest that has never been described before, your job is to give it to somebody who can name it, or an expert who can name it, and you're going to give it a name. But it's not a single name, it's a two-part name. So all discovered species, whether it's a plant, an invertebrate, a virus, an insect, a crab, whatever it is, gets a two-part name that's called binomial nomenclature. And that's kind of what this guy came up with, and he started naming organisms a long time ago. And the two-part name is the genus and species name, okay? And so these two are always italicized, and the genus is always capitalized and the species name is always lowercase. So here are two great examples. Here is a manta ray and a concentric ray. All right, so you got Manta hamiltonii after named after a scientist, and here you got Leurophoros concentricus, which is the concentric ray, and you can see the rings of color around it. So names, you get to name it, right? The name that's unique to all organisms is the species name. So the species name is the name that you see will have a lot of variation. Every species fits into a genre, all right, or a genus. If it doesn't fit into a genus, guess what? You get to name the genus also, okay? Which probably means you'll be in a new family if that genus doesn't fit into a particular family. So why do we do this? Because we are reductionist, biologists are. Naturally, we like to break things down into smaller units so that they're easier to study, right? So a group of taxonomists that blow my mind all the time whenever I hang out with them, because I have several friends and colleagues who are bony fish taxonomists. And I've been to the Amazon with them. And we'll throw a net in and they'll pull a fish out and they'll start naming all these fish. It just blows my mind. And there are over 20,000 species of osteichthyans, which means bony fish. And so you can imagine that's pretty daunting. So it's much easier if you break them down into groups, right? And study specific groups instead of trying to study that entire group. Same thing goes for arthropods. There are millions of species of arthropods. Can you imagine trying to study those? And I've been with Dr. Milne before. He's phenomenal. We'll go to Costa Rica. He'll find a spider and be like, blah, blah, blah. And I have no idea what the hell he's talking about, right? Because he knows the genus and species of all the spiders. There are 45,000 arachnids. So I'm just going to stick to histology. I'm not, this is a little bit much easier. Now, there is a classification and phylogenetic hierarchy. Really, classification or taxonomy, there's this hierarchical kind of grouping that is very common. And you've seen this before, right? And so we got kingdom, phylum, and we even have domain now, right? You see domain, domain eukarya, domain prokarya. But we're going to just stick with kingdom. So we got kingdom up here, and since we are dealing with just vertebrates, basically the first three names are always going to be the same with any animal we look at taxonomically. And basically the way the naming system works, if you don't remember, is we start very generically and end very specifically, right? And every group fits into another group if we do it correctly. And here are my favorite vertebrates. So this is a dart frog. As a matter of fact, I have that dart frog downstairs. This is Dendrobates tinctorius, which means the painted frog, or the dyeing frog, like dye color, has been splashed on its back. And tinctorius has a bunch of different morphs to it. The one I have downstairs is called the Patricia morph. So my frog downstairs has lighter legs, or like light blue, not dark blue. But this is a frog, so it's a dart frog. And so you can see here's its unique name. Dendrobates is the genus it sits in. Tinctorius is the species name. You can see the genus is capitalized, the species is lowercase. These two are italicized. Now, every genus fits into a family. So you can see here's the specific species. Every species fits into a genus, right? And the genus can be small, 3, 2, 1 animals, or big, like 90 organisms. Just depends on how diverse the vertebrate we're talking about is. So I study a lizard called Scoloporus, and Scoloporus has 90 species in it. So that's that's a very speciosus genus. Where you could look at, there are a couple smaller lizard groups, genera, that have 2 or 3 species in it. Dart frogs, Dendrobates is a pretty large genus, so there's a lot of dart frogs in that particular group. All dart frogs, because there are 3 different genera, sometimes people say 4, sometimes 5, fit into one family. And so you take all the genera and stick them into a family, which is kind of the next larger group, and every family name ends in DAE. So if you ever see a taxonomic name that ends in DAE, you know it's a family of something. So Dendrobatidae includes all of the genera of dart frogs. Now, frogs, it's a type of frog, right? So all frog families, and there are lots of them, there are like over 8,000 species of frogs now. So a lot of frogs. All the frog species live in an order, the next group up, right? Which is Anura, a great Latin name, Anura. And Nura means tail, A means without. So without tails. All frogs lack a tail, except for one group, one family. So they're tailless. And then frogs are an amphibian, right? So amphibians, all orders of amphibians belong in the class called Lis amphibia. Lis means modern or living, extant. So this would not include some fossils, not all fossils, but a number of fossil groups. And frogs and amphibians are just like other birds, reptiles, and mammals. We have backbones. And so we're going to throw all the classes of these backboned animals into vertebrata, which is the subphylum. And then all vertebrata belong to the larger phylum Chordata or Chordata. And then chordates are one type of animal, right? There are many types of animals. So notice with phylum and subphylum, you can put sub and super and infra. There are a whole bunch of subcategories that people are now sticking in, because what we're going to try to do is take our classical taxonomy and try to figure out if those groups are phylogenetically or evolutionarily sound. And if they're not, then we've got to move animals around. And sometimes we've got to add subgroups. So notice we did that with this one here. Not all chordates have a vertebral column or a backbone. So we had to put them in their own phylum because it's monophyletic, which is vertebrata. We're going to talk about what that means here in a second. So this is kingdom through species. And you can, in every organism you look at, has typically been able to fit it in. Now, I will tell you, in the Smithsonian, there are animals and invertebrates that are so weird that we have no idea where they belong. And guess what? They don't have a name. They still don't have a name. People are still trying to figure out where they fit. And if you're weird enough and you get your own genus, often you end up with your own family that then goes into an order. But I've even seen sometimes there's things that are so bizarre, fossils included, that end up getting their own order. And that's pretty substantial because they are completely new to science. And that does happen. All right, so what's the difference? We want to know the difference between kind of classification, taxonomy, and phylogeny. So phylogeny, think of it as a family tree, right? And we build a tree, looks like this, right? And it has a bunch of branches on it. And then all of these organisms are sitting on top of one another. And you can trace them back to family members, right? To true evolutionary ancestors. We're going to call them ancestors. And if you notice, just like any tree, there's dichotomous branching here typically. So one species splits into two, right? And that's how it typically works. Now, that's not always the case, and we'll talk about some special occasions that happen. Now, you can see, you can see different types of groupings of organisms that they call true phylogenies. And some of them are really simple, like this one, and then some of them are more complicated, like this one. Okay? What we don't want to do is think about animals moving in the direction, or since we're just dealing with vertebrates, in the direction of mammals. In particular, you don't want to see humans at the top of your list of organisms that, all vertebrates move to mammals, and mammals are the most derived, and we all move to humans. Humans are the top of the chain of evolutionary dominance, if you will. I want you to think more like this down here, where mammals are on one side and reptiles or fish or lampreys are on the other, and they've all undergone their own very complicated, very old line of evolution, right? And so mammals are sitting up here as the apex of their evolutionary line. So are the modern living organisms that are in any other group, right? They are really specialized, highly adaptive organisms that fit their environment really well, just like all other mammals do. So this is called the ladder effect, right? Which was put into the heads of the educated, back with Greek philosophy, and is stuck with religious, obviously, right? Influence suggests that humans are the epitome of the creation of God, right? So we don't want to think of it this way. We want to think of it more like these are all independent lines of evolution. Does that make sense? And there's a lot of organisms that are obviously extinct, and only the ones at the top of the phylogeny are going to be living organisms. This is completely wrong. This animal gives rise to this animal, gives rise to this animal. None of these animals are direct descendants of each other. This is more the ladder effect. I pulled this out of a non-majors biology text. It's pretty funny, right? So this is completely wrong. It's just really oversimplified, right? Which is terrible for a non-majors text or people that aren't scientists, right? So the other thing that you often hear is lower and higher, like lower fish and higher mammals. Again, that gives you this discrepancy. I like primitive and derived, or ancestral and derived. Basal or derived is a little bit better terminology. That's not so different between the two groups. Lower or primitive simply means that they show more primitive characteristics that are older characteristics, where derived show more recent changes to body form. But you'll see them in text, you'll see them in papers. They use lower and higher, primitive derived, basal derived. So this is the hierarchical scheme of classification. And it's pretty, it looks nice, right? And you can trace these all the way back to phyla and kingdoms. Phylogeny is more interested in tracing evolutionary relationships, not necessarily that you look the same. So here's what classification looks like, right? So if you could trace it back, the role is to group organisms based on similar characteristics. So you have all these genera. They fit into 4 different families, into 2 different orders, and 1 class, right? So phylogeny is going to look like this. It's going to look maybe similar to it. It's kind of a branching system. And if we actually named all the organisms right, based on their common ancestry, the phylogeny and the classifications would match up really nicely, like this. And they do sometimes, but often they don't, because 2 animals that look alike aren't necessarily related, and 2 animals that look really different are sometimes closely related. And so that's what really throws off classification and phylogeny, because they group organisms on different criteria, right? This is what it comes down to. Now, phylogenetics, or the grouping of organisms based on their evolutionary past, is also called cladistics. There's another term that they use. Cladistic classification is phylogeny. And we're going to talk about how you do this in a very simple way. Most phylogenetic trees are now built using supercomputers. All right, and if you've ever built a phylogenetic tree, and I'm sure all of you did in 155, we had an exercise where we built a tree, and maybe you had 10 or 12 or 13 characteristics, and it was hard, wasn't it? So imagine trying to do hundreds of thousands of characters. That's why you let a computer do it, right? So now, the one thing I always want you to remember, any tree that you see is a hypothesis, right? What does that mean? That means you could prove it wrong, right? I know prove's a strong word, but it is just a hypothesis. It is the best educated guess that we have with available data, right? So trees change a lot, and that is pretty annoying, actually, right? Especially if you learn one tree, 2 years later it changes because of new data. So it is a hypothesis of evolutionary relationships. It's based on the information present when it was built, And if you find another species of turtle and throw it into the mix, it could change your tree completely, right? And then you would republish it. Now, on phylogenetic trees, new species can form in two major ways. The most common way is called cladogenesis, where a species breaks up or diverges or splits into two or more, but typically it's dichotomous, two. Anagenesis is something that's not necessarily more rare, it's just you don't always see it. It's when a single species simply evolves into another one. There's not two of them. And it happens through mutation usually, or some weird or odd circumstance. So we form these branches. There are a couple, these are the two major ways to represent them. I really like this way because it just makes more sense visually, but a lot of trees look like this, where you have this line and then branches coming off of it. You can add branches to the lines as they go up. And typically time moves up or across from left to right in these trees. And organisms that are close to each other, they share an internal node or branch, are closely related, while those that share further away and further and further and further away branches are more distantly related, right? So you can see here A and B are very closely related because they share this common ancestor E, or these suite of characters E, that nobody else does, right? And these are called sister taxon because they are next to each other. A and B are more closely related to C than D because they all three share this common ancestor or suite of characters in a common ancestor. D is at the base of our tree here and is our outgroup because it will share this character with all the rest, right? And you could continue this if you had more species. Same tree this way, just the branching is different, right? The more characters you add, the more robust the tree gets. The more species you add, the more complicated the tree gets, right? So far, so good? Okay, the tips of a phylogenetic tree represent taxa, or groups. You guys have all heard the word taxa, right? I use it a lot, taxa or taxon. Taxa is the plural, taxon is the singular, and all taxa means is group. All right, so a group of organisms that is alive, or the living descendants, are what sit at the top of a tree. Now, these groups could be individual species, they could be genera, they could be families, right? It just depends on who's doing the phylogenetic study. The nodes on the inside are where the branches meet. Those are called internal nodes, and they represent common ancestors of those descendants. And when I say common ancestor, sometimes you just see a list of traits. 4 legs, anucleated red blood cells, hair, mammary glands, eyes, whatever it is, right? Those traits are the ones that are grouped together in the ancestor they share. Sometimes they might be called derived characters too, or ancestral characters, and we'll talk about the differences here in a second. Notice if we do this right, our trees are rooted, right, somewhere, with a common ancestor that gave rise to everybody. Now, this ancestor is not always known. Depends on the group that we're talking about. The length of the line often represents time. So you can see the shorter the line, the least amount of time between these 2, 3, and 4 diverging from one another. That's why they're closely related, right? Notice number 1 in this particular phylogenetic tree is the oldest. It has the longest line, right? So, for example, this is where fish would be. Fish are a really old group. This is where mammals would be, up here, 3 and 4. It's a more recent group, right? We're young. We're a young group. Derived, very derived, but very young compared to, say, fish. So here is a bird phylogeny from a bird textbook. And there's a whole chapter on phylogeny in this bird textbook, the ornithology textbook. But this shows you really good what we just kind of talked about. So these are the living guys up here, right? Usually, so in the present, you can see there is a timeline here, present to about 10 million years ago. And so these birds here that have been drawn that don't make it to the top are all extinct, okay? And you can see when they lived, based on either there's a fossil or there is if they're using DNA and RNA, you can use mutation rate and compare differences in a gene, like mitochondrial gene, and you can estimate when the organisms diverged. And it's a really good guess because often if you have fossils and DNA, you can compare the fossils and DNA and you get similar types of numbers, all right? So it's not always the case that you have that, but there are some cases where that has been possible to do. And what we call our molecular timeline, right, or the genetic clock, if you will, is the best guess we have for divergence times. In a lot of animals, we don't have good fossils for it. But you can see here that we have basically dichotomous branching, right, going along until you get to something like this. See where D doesn't branch into something else? The D organisms give rise, bird here gives rise to a G through some probably mutation or a suite of mutations. And now G, which is a completely different species, gives rise to G and J, right? So that's anagenesis. It's another anagenic event right over here. But mostly you see dichotomous branching to give rise to the modern birds that these are songbirds up here that you see. So like cardinals and orioles and finches and so forth. Okay, so far so good? Okay, monophyletic groups. These are true phylogenetic groups, or we call them clades. Clade is the taxon name of a monophyletic group. Don't you love anatomy? Anatomy is the worst discipline. There are like 50 names for the same thing. It is really annoying. I didn't come up with it. I have to teach it, and I had to learn it too. But a monophyletic group is a sound group. You can trace all of the descendants back to a common ancestor. That's what phylogeny is trying to do. When you see a tree, that is our best guess of the monophyletic groups at that time, okay? Or the clades, birds, for example, which we just looked at. So it's a group of species or some other taxon consisting of the most recent common ancestor and all of its descendants. Smaller and more recent clades, like mammals, right, are subsets of larger clades. Mammals belong to amniotes. Amniotes belong to vertebrates, right? So the clades get larger and larger and larger. The larger the clade, the older it is, right? The smaller the clade, the more recent it is. So, for example, when you think about classification, if I try to think about what is going to be the most recent taxonomic hierarchy that'll fit into a tree, it's going to be the species names, right? Or the genus. The genus is going to be, if it's done right, will be a more recent representation of the animals of interest, whereas a kingdom is really old, isn't it? Can you imagine when the first animal arose? I mean, it was a long time ago, right? Or the first reptile? I mean, it was hundreds of millions of years ago. 500, 300 million years ago, something like that. So, whereas a lizard, a modern lizard, which belongs to a genus, is a more recent event, right? Something like 50 million years ago or 10 million years ago. Does that make sense? And I'll show you a visual representation of that right here. So, here's a phylogeny. And when it's possible, and we're trying to do it in some cases, but in some groups it's impossible. You got your kingdom down here, which notice there are 64 species within that kingdom. This is hypothetical, by the way, right? If we could do it correctly and put taxonomy on top of phylogeny, you can see the oldest group is the largest taxonomic classification. The most recent groups are the species in the genre, right? They're closer to the top. They're also the smallest groups, right? The more specific groups. And as you go down, you can see that they kind of all fall into place. Now, so where's all the controversy come? Most of the controversy between classification and phylogeny happens up here, right? The more recent breakdowns, because a reptile was always a reptile, right? I mean, so these are big, huge characteristics that fit all reptiles, both phylogenetically and taxonomically. So where we fall into trouble is sometimes we'll throw a particular species in a genus that's not evolutionarily sound, and we got to make a new genre so that those frogs that we thought were in this genus really aren't. We have to split them, and then now you have a new genre, right? Sometimes you might even have a new family. It just depends on the group. And typically, most of these original groupings of organisms were done by what they look like, right? They're what we call phenotype. So we use skeletons, we use features that we can see. Now, when you start messing with their genetics, though, and you start drawing genetics and looking at DNA sequence, which is pretty rigorous and pretty sound, sometimes two things that look alike, like we mentioned, they don't share the same genetic similarity, and so we have to put them in a different group. Reptiles and amphibians are a great example of this, and so their names are changing all the time. So how do we do cladistics? So how do you make one of these trees? Yep.
For the last slide, so there's 6 monophyletic groups.
Oh yeah, within there are, yes. Yeah. And so you got big ones and small ones, right? Yeah, you're right. Good. So how do we make these? Well, you need a database, right? You need something to measure, right? We're doing science here. And really, it can be anything as long as you can convince reviewers when you're writing a paper. So how have I done phylogenetics? I've used sperm ultrastructure because I can build a huge data set out of sperm ultrastructure. And I can collect that data from multiple species, and then I can compare, those things. So these are some examples of things that have been used in the past to build phylogenies. So you can use DNA and RNA. These are the two most sexy things to use now, right? The most accepted. It's the best way to think about it. Everybody thinks DNA and RNA is the best way to do phylogenetics. I'm an anatomist, you can imagine. I'm kind of biased. I don't like molecular biology that much. So the classical way to do it is morphology, right? That's what I like, right? Morphology, anatomy. That can be whole organism morphology, or it can be histological characters, right? Or you can use amino acid sequence of proteins. That's another good way. You can collect proteins and look at them. You're comparing the same protein, like keratin. Or you're comparing the same DNA sequence, like the mitochondrial or the cytochrome C protein. You could use behavioral characteristics. I've seen some trees built on how organisms behave. Well, that's pretty unique, but it's been done. And so if you can get a big data set, then you can start using statistics to help you understand the relationships between them. And computer programs or softwares will then build trees for you. Now, all these trees are going to be based on something that's called parsimony. And the data analysis that they use is really it's high tech, right? I'm not a statistician. You can look up Bayesian statistics. It is not kind of easy to understand, right? Maybe unless you're mathematically inclined. There's a whole bunch of different types of statistical analysis that you can use to test whether a tree is sound or not. Bayesian stats happen to be one of them. Parsimony is easy to understand. Parsimony is the assumption that the simplest tree is right? My dad used to say, keep it simple, stupid, right? The simplest tree is the most correct tree. That's what parsimony is. I'll show you a slide of that in a second. So once you say, all right, I'm going to collect these characteristics, then we are going to compare them, right? Then we're going to compare them. And what we want is we want to be able to group organisms based on their derived characters. All right, and I'm going to define what a derived character is in a minute. Derived characters have a sciencey name, all right, a scientific name, biological name. They're called synapomorphies because shared means syn, apo means derived, and morphes mean characters. So synapomorphies are the characters that are going to separate our organisms of interest into groups. Okay, they're the branches. Synapomorphies are at the branches, okay, of our tree. But the problem with that, that's great because we think about similarities, right? But that's good, but the similarity is not enough for cladistics. We need to have it reflecting descent by modification, right? We got to be able to trace them back to a common ancestor. So not only do you need to gather characters that separate our groups, but we need to know what the ancestral characters are, right? How do you figure out what's ancestral and what's derived? That can be easy if you have a good fossil, or it could be impossible, right? If you don't have a good fossil record. But, you know, those are the problems with phylogeny. There's problems with everything. So once we've kind of understand what's derived, what's ancestral, and we think we can do this, then we got to give character states to our characters, which simply means we want to keep it simple so that our data analysis program can actually do something with it, right? So if we use eye color, then we can use blue, red, green. You can use an actual quantitative characteristic. If we're going to use like mammary glands, then they're either present or they're absent, right? So you can give them 0 if they're absent and 1 if they're present, right? So you do that with numbers of legs. You could just use 0, 1, 2, 4, 6, 8, 10, whatever it is, number of legs. So you're going to give them some kind of characteristic that we can use, a character state is a better term, that we can use, and we then can then give them letters or numbers that the computer can then start matching them up, right? And that's what the software can do. Once you throw all those into a computer software, then the software will ask questions like, well, what's an apomorphy? Which of these are apomorphies and which of these are plesiomorphies? And you'll get different tree outputs depending on, you know, your designation or the accepted designation. So plesiomorphy means old school character or ancestral character. So a primitive or ancestral character is a plesiomorphy. If a group shares several of them, then they're called symplesiomorphies. Now, everybody ignores these characters when they teach phylogeny, which I just don't understand. Symplesiomorphies are really important because they define your clade, right? They define your clade. So if I wanted to put all reptiles on this planet in this room, how do I know it's a reptile? All reptiles share the same characters. They have to be ancestral for that group, right? Therefore, I would use those symplesiomorphies to define my clade. I throw all the reptiles in this room. Can you imagine if we could do that? That would be sweet because I'm a herpetologist. I like reptiles. If we could put them all in this room, that doesn't help us very much though, does it? We just now have all the reptiles in this room. So how are we going to study them? Well, we want to break them down into smaller usable groups, digestible groups. That's when we use the derived characters, the synapomorphies, to start grouping different reptiles on the tables into different groups. Right there, and we group them until they get to smaller and smaller and smaller and smaller groups, and then we have them in 1s or 2s, right? If we do that by eliminating traits that some have or don't have, and that's kind of what a computer does, right? So we use the symplesiomorphies to determine if you're in the clade or not in the clade, right? We call it the in group or the out group, and then once you're in the in group, we got to put you somewhere. And then we use the apomorphies, synapomorphies, to do that. Okay? Now, every once in a while, a character will be completely unique to that organism only. It's not found in any other organism ever, or at least presently. Those are called autapomorphies. And so if we had one reptile that just had horns that were made of silver metal, which doesn't exist, that would be an autapomorphy, right, for that animal. So branch points on our tree, two species differ in a shared derived character, right? If you're in the in group, then you're part of the monophyletic group that were of interest, okay? So we could use plesiomorphies to do that. If you're not in the group, but you have maybe one or two of the ancestral characters, for example, and not all 10, then you might be the closest related species to the monophyletic group. They're called our outgroup. Okay, the outgroup roots our tree or helps us understand what's ancestral versus what's derived. If you don't have an outgroup, it makes it really hard to know what's ancestral and what's derived, right? And your tree won't be rooted. It'll be circular. So if you look up a bacteria, a true bacteria phylogeny, it looks like a sun that all radiates to the middle because they don't know what the features of the first bacterium or archaean or prokaryote was. And so we don't have a root. So one of the first things you have to do to build a phylogeny is determine who your outgroup is. And so how do you do that? Well, you can go into the literature and look, right? If you want to study a group of lizards and you might want to know, well, what's the outgroup lizard to this group? You can go into the literature and look that up. So I've done that. So remember I said Scoloporus, this was a big genus of lizard. You don't have to remember the name. And when I started studying sperm characters in that group, because I was just really interested in that group, I needed an outgroup, right? I didn't have one, so I have never been able to build a phylogenetic tree that's sound. And I just recently did a study in Phrynosoma, a different genus that is considered ancestral to Scoloporus. So it would be a perfect outgroup, right? Because it's considered older or not in the same group. So they would be a perfect group. They sit right next to each other. And I could use maybe some of the traits that the Phrynosomids have, that Scoloporus have, to say, oh, look, they are at least somewhat related. But all the Scoloporus have these apomorphies that separate them from synapomorphies, right? From Phrynosomids. And now we can then look at those 90 species of Scaloporids and break them into smaller groups, which we would call species names, right? Does that make sense? So for vertebrates, remember I showed you phylum Chordata, subphylum Vertebrata. For the vertebrates, the organism that is often used as the outgroup is a little chordate called a lancelet, right? Or Amphioxus is the name. You'll see that on your lab handout later. So this guy's used because it has a lot of the characteristics that vertebrates have, but no backbone. All right, so it's missing one of the, what we would call synapomorphies of vertebrates, right? And so it makes a great outgroup because it roots it right in place and shows you that the spinal column is a good apomorphy, right, for the group of vertebrates. Now, if you're just talking about vertebrates, though, then the spinal column is a symplesiomorphy, isn't it? So it just depends on how you use your characters. Okay, so here's an easy or small phylogenetic tree that kind of helps you, right? If we say D is kind of our outgroup here, you notice that blue and square is plesiomorphic then, right? If we're going to call this whole thing a clade, right? So this is a clade. So notice, and if I tell you this is the primitive side and this is the derived side, you would figure that out by just looking at it. But if you look at D and C, notice D and C are blue and D is square. So that square is the D is the only one that has a square. So that is the plesiomorphic condition, and so is blue, right? Small size is apomorphic for A, right? It's the only one with really small size. Or you could even call that a topomorphic if you really wanted to, right? Because at least if these were the only animals that we're looking at. But the circle is a synapomorphy for A, B, and C, isn't it? right? Because it separates them from D. And then obviously red is synapomorphic for A and B. All right, and then A, the small size, is apomorphic, which separates it from B. See, so you basically are eliminating traits as you go from right to left. Make sure I'm got my direction right. So there's polarity, right? Polarity to your tree from base to top. All good? So we've already kind of did this. All species are placed on the top. Each cladogram is a hypothesis that shows shared derived characters as you move up and separate groups out. There's an outgroup to determine plesiomorphies. And we're going to choose the most likely clade based on parsimony. Okay, that is the fewest steps. So here's a great example. Here's one clade of fish. These are cichlids from Lake Victoria. So they're using these characteristics. Notice it takes 4 on this one. Woo, whoops. And 5 on the other one, right? So there are 5 steps on this tree and 4 steps on this tree. So the most correct one for us is the simplest one, the 4-step one, right? So this is the one we would accept because it's most parsimonious or the simplest. That we do that automatically, right? That's what all software does. We got to make some assumptions somewhere so we keep it universal, right? Because you can imagine once you start getting hundreds of thousands of characters, can you imagine how many trees will be spit out? I mean, you could spit out any tree at that point. You want to spit out or limit the software to make a tree that takes the least number of steps, which is the most probable way that the relationships are built. Now, the biggest problem with phylogenies is convergence, as you can imagine. Sometimes we group animals together because they look alike, and we're like, ooh, these guys are related because they look exactly alike, but they're not, right? Obviously, these are extreme examples. Obviously, dolphins aren't fish, but you often hear people call them fish, don't you? That aren't scientists. I mean, they look like fish, kind of. Not really, but they are fishy, like as far as their fins go. But that's a really good example of a very specialized environment where the environment is more important in determining what you look like than your ancestry, right? Same thing with wings and flight. The environment really shapes the structure of the wing more than your ancestry does. So when a dolphin and a fish, these were grouped together by Linnaeus back in the day. Anything that had fins was put into the fish group because they all had fins. And these are mammals, man. They're more closely related to Linnaeus than they is to this animal. This is a really old group of fish. So when this happens, when we mess up, it's called an unnatural clade. It's not a big deal. That's how science works. Because remember, every tree is just a hypothesis. If you figure out, because of new data, that 2 animals are convergent, then you simply publish a new paper. It's great. Right? And phylogeny. And we were talking about convergent evolution, modified. Yes?
Do you want your microphone?
No, I'm going to go with that. Thank you, though.
You're welcome.
We were talking about convergent evolution, homoplasy versus monophyletic groups, which are true clades, right? Where we can trace all the descendants back to a common ancestor. And here's a really good example of that. A lot of times it's real easy to see, right? Like a dolphin and a fish, and you're like, ooh, that's convergent evolution. 2 really different groups. But it's a little harder to do in animals that are more closely related as far as where they come from. So, for example, different types of cichlids. Okay, so these are cichlids in 2 different lakes. Now, what's interesting about most of the really large lakes in Africa, during the history of the development of Africa, these lakes flooded and dried out and flooded and dried out and flooded and dried out over millions and millions of years. And cichlid evolution is incredibly explosive. So it's very, very explosive. It undergoes kind of a process we call punctuated equilibrium, which we're going to talk about at the end. It's the last slide of this lecture. And when you look at these two lakes today, you will find fish that look really similar to one another. And what's really interesting is when they started doing these genetic studies on them, they thought, oh, these are related because they're really closely related probably because these lakes flooded a long time ago, or recently, I should say, and then separated by drought. And when you look at these two lakes, this lake, Malawi, is about 1 to 2 million years old, right, as far as being separated from all the others. This lake here on the left is much older. It's like 12 million years old. And when you look at the fish from both lakes and compare them, you can pair them up like this. And when they were morphologically put together, obviously, as a morphologist, you would say, oh, these two are more closely related than everybody else and so forth, right? But when you look at the actual genetics of each individual lake, all of these fish in Lake Malawi, they form a monophyletic group. You can actually trace their genetics really nicely back to a common ancestor, which was one type of cichlid, actually, that came from this lake, actually. So that's kind of cool, right? And because it's an older lake and it has lots of older lineages in it. And so when you look at these, all of these arose from a different common ancestor. So they're not monophyletic. They are polyphyletic in the origin of, and we're going to learn what polyphyletic means in a second, from the origin of their ancestors. So they're not related to each other, and they're not related to the guys in the other lake like it was once thought. And so this is an example of homoplasia because the environment that these two fish occupy is similar. It's a grassy environment. It's an environment where their stripes break them up, and so they escape predation. And these two are alike because they're predators, or I should say these two are predators. These two are alike because they're mouth brooders. So the environment is shaping their similarity. And you can say that because that's kind of how evolution works in these two instances, because these guys over here formed from what you would consider normal Darwinian evolution, where these guys have all kind of mixed lineage as far as their ancestry goes. And so this is a really good example when you start comparing fish across both lakes of a homoplastic convergent relationship. Does that make sense? So hopefully that's a little better than what I did yesterday because I did it too fast. All right, so I said monophyletic and polyphyletic. So what does that mean? And so there are kind of three branching patterns that you can see in a hypothesis of a tree. So remember, make a tree, it's a hypothesis. I try to present it as a correct tree. When I'm presenting, or what my null hypothesis is, when I build a tree, is that the groups that I show you are monophyletic, right? I can trace them back to a common ancestor. But sometimes what I thought was monophyletic is not when I get new information. And when that happens, then the branching pattern can be of two other kinds of ways. And they both start with P's. And that messed me up as an undergrad a long time ago. They're called paraphyly and polyphyly. And the name is pretty nice. Phyletic means lineage. So a mono lineage, one lineage, right? Poly means multiple lineages, multiple common ancestors. And para means without. And so you're missing lineages, right, with your groups. So remember, a taxon is just a group of animals. And a clade is what we're trying to group them as, right? Our taxon, we're trying to make a clade. And a clade is a monophyletic group where you can trace all the descendants back to a common ancestor. And typically, we're always, because we're studying vertebrates, going to see rooted clades where we have an outgroup that roots our tree so that we know what's primitive versus derived, like we talked about last time. So let's look at the differences between these. This is out of an evolutionary text that shows you monophyly, paraphyly, and polyphyly kinds of groupings, right, on a tree. And so you can see here, when you're looking at this tree, if I concentrate just on the groups from C and up, this is a nice monophyletic group, isn't it? I can trace L, M, N, and O straight back to C, right? So if this is my group of interest, then I have a common ancestor C that gave rise to all of these organisms over time, right? So it's a nice monophyletic group. Now, in a paraphyletic group, I might pull A and B into my study the next year and say that this is my new monophyletic group. And that might be true at that point until you start getting information about these other species. And when I get information about these other species and realize they belong in this clade, then my original hypothesis is paraphyletic because I forgot these groups. Does that make sense? So, good example of this where we actually do it on purpose. I'm a herpetologist. I study reptiles and amphibians. And if I just focus on reptiles, I do not include birds. That's paraphyletic. Birds are reptiles. But I don't study birds. I just study reptiles. And I make all my assumptions on reptiles. And I'm making assumptions that birds follow the same type of histological information, for example, or architecture. And that's not always true. So a lot of herpetologists don't include birds in their discussion. And that's really paraphyletic. So when I teach herpetology, I'm teaching herpetology next spring. Didn't know that. I'm going to, for the first time here, I'm not going to talk about birds. We have an ornithology class for that, right? Dr. Sweets teaches that. Someday I might teach it when he retires. But I am going to teach a herpetology course next spring, and we're going to just focus on reptiles and amphibians of Indiana and in general. So that's paraphyletic. So if you see the original tree is paraphyletic in a paper, then you'll be like, oh, okay, they forgot some groups, right? That we now know exist or belong in that monophyletic group. Polyphyletic is what happens with convergence. So polyphyletic often equals convergence because what we originally thought was monophyletic, like those fish, actually is not, right? And so you have more than one common ancestor. So if I say all of these are my monophyletic group, you start trying to trace them back. This side works okay, but L and M don't go back to B. You see that? And so they go to C. So you got two common ancestors here, and that's polyphyletic. Now, if I go all the way back to A, then I'm okay, right? So those are the types of groupings that you can see: mono, para, and polyphyletic. Yeah. Poly is, it's considered wrong because it's not truly monophyletic. So a lot of the old classical names that you use, like Amphioxus, for example, which I'm going to teach you, is the only living representative. It's not a monophyletic group because there's a bunch of fossils we don't include. It's actually polyphyletic. And so we're always trying to publish hypotheses that are monophyletic in our mind, but they don't always work out as we gather more and more data on a group of animals. Does that make sense? And it's convergence that does it, because we often group things based on morphology, right? And what they look like. Okay, here's a great monophyletic example that's pretty universally accepted. This is a tree that involves starfish, or what is called Echinodermata is the group name. And so you can see it forms these beautiful clades that are supported by DNA, RNA, protein, and morphological studies. So it's really robust. It took a while to work it out, actually. So this is a great example of that. So there's a bunch of different organisms in here. Now, when you look at this, notice you got Echinodermata is our big group. And these are all the echinoderms, at least on this study. And here's my outgroup. Right, so this is my outgroup over here. It's called Hemichordata. Now, the reason I point that out, because you really probably don't care, right, is that Hemichordata is not necessarily primitive to this group, right? It's actually because, I kind of say you use an outgroup to root your tree, and you probably immediately assume it's ancestral, right? It doesn't have to be. You can use a derived group above it as your outgroup, right? It still roots your tree, and hemichordates have a few of the echinoderm traits, particularly their larvae look a lot like an echinoderm larva. And so they're probably pretty closely related. And so we're actually using an organism here as our outgroup that's more derived than the echinoderms in the sense of traits that it carries. But it's a nice close relative that we can use to root our tree. So this is an old phylogeny. Now, I guarantee if you went up and looked up echinoderm taxonomy or phylogeny, this tree would have changed a little bit. That was a long time ago, right? This is an old example. Here's the classical setup of reptiles. So when you look at reptiles, now this group here, these are called amniotes. All right, so these are called amniotes, this really large group, which is a monophyletic group. Here's the job. So I've been lazy and not writing words down, right? So amniotes. So amniotes are, can you see that? Jesus. So amniotes are a group of vertebrates that have extraembryonic membranes around their embryos. And they include birds, reptiles, and mammals. Okay? So this is a tree. And I ask you this, the classification of reptiles and birds. Are birds monophyletic with the reptiles? Or another way you might see this is, are birds nested in reptilia? And when you look at this phylogeny, notice they are, right? So birds are right here. And look at our outgroup, mammals. Right, so mammals are our outgroup, and everybody else that's an amniote is a reptile. Right, so really, if you start talking about reptiles, you should not exclude birds, right? Or if you talk about birds, you shouldn't exclude reptiles. But ornithologists and herpetologists disagree with that. Of course, we will separate them. I love birds, though. Actually, I'm a big birder. I'm a bird nerd, if you will. I like them. Right? Photograph them a lot. But so they're here, and they're really closely related to dinosaurs. Birds are the modern dinosaur. They're the living, extant representative of the dinosaur line. They have direct descendants from dinosaurs. So these are the rest of the reptilian orders, or I should say not orders, reptilian taxa here. And we'll go over different kinds of them. This is not a reptile. I should put a reptile on there, huh? That's a salamander. And there's a lot of textbooks that are on just ornithology, a lot of textbooks that are just on reptilia. And when this is where things get iffy, right? So, if you start talking about a taxonomic clade and you call it reptilia, it's not really a true clade, right? I mean, so you can't say there's a reptilia and aves. Aves are birds. Can't do that. You're going to have to call them reptilia, and then you're going to have subgroups under those, right? So you start adding subgroups to those. So they're going to be called subclasses instead of a class. So if you see class reptilia and class aves in a textbook, they're using those as, what the heck? Oh, it wants me to get on the internet. Took it long enough, didn't it? It wants to, it's just a means of convenience, if you will, for whatever we're talking about at that point. So that screws things up. We have to include birds in if we're going to call this a true class. If not, we're going to have to give it a different class name and then give it subclasses, which is what's basically been done. So if you're going to do true monophyly, you call it class reptilia, and then subclass aves, and then you'll give different names to the reptilian groups. The other crazy thing, I'll just show you, because reptiles are a mess, is you see where turtles are way over here? This is how they're classically thought of, turtles. Very the old group. Notice mammals are a really old group, right? Remember, line length gives you a kind of age. Turtles might belong right over here. They might be sister to birds. And that's what the genetics is saying. And so it's going to, once that, it is pretty universally accepted now. This phylogeny is completely wrong, right? There are still some conservative herpetologists that say turtles are primitive. They're ancestral, but, they really aren't. What's really interesting, I study sperm morphology. The sperm of a turtle looks just like a bird sperm. Doesn't look like a squamate sperm, like a lizard or a snake sperm. It looks like a turtle sperm, or it looks like a bird or a sperm, and I assume what a dinosaur sperm would look like. So, my studies suggest that turtles are sister to what we call archosaurs, is the name you'll see when we start talking about reptiles. Okay, so here's another example. You might see two foxes and be like, oh my gosh, they're so cute. They're really closely related. These two species are very far apart within Canidae, which are canine carnivores. So you can see here's the red fox up here. This is the fox you see here in Indiana, and this is the gray fox. This is you see this in Africa. Convergence, right? They occupy very similar habitats on two different continents. And they look a lot alike. It happens a ton, right? It happens a ton until you look at the genetics. And it makes sense. This fox and its ancestors are North American. This fox and its ancestors are African. Mammals, when Pangea was around, mammals were really little. There were no foxes around. There were just dinosaurs. So once they separated, they come from different ancestral lineages. Now, the last thing I really got to cover here is kind of mode of evolution. So another thing that a lot of the general public doesn't like is that biologists can't make up their minds, is what they say, or they argue over how evolution happens, right? And so they get caught up on that point. Evolution is real, right? It's common sense. You can see it in everyday life. We do argue over, what we call the mode, or how fast it happens. Classical Darwinian evolution is gradualism. So we call this Darwinian gradualism, Darwinian evolution. We're talking at the macroscopic level, right? So this is macroevolution, speciation. So gradualism is when you get all these small intermediate changes, and it takes really long periods of time, right? You need hundreds of millions of years to see the diversity that you see. Punctuated equilibrium is really fast, like those cichlids, right? All those cichlid species that arose within Lake Malawi, that happened within 1 million years. That's really fast on an evolution time scale. And that's a really good example of what they call punctuated equilibrium. And the guy who came up with this is dead now. His name is Stephen Gould. And what's incredible, this guy is brilliant, by the way, is he came up with this as a grad student. Can you imagine trying to come up with something as a grad student and then defend it against all those old stuffy PhDs, right? And so he came up with this a long time ago. He was a Harvard professor for a long time. And what punctuated equilibrium says is that you're going to have periods of stasis in the fossil record and then explosions of diversity. And that explosion of evolution, it's called adaptive radiation, happens when large niches open up. A bunch of niches at once open up. All right, so a great example of this punctuated equilibrium is when the dinosaurs all died out 65 million years ago. Mammals were set and ready to go to take their places, right? And so immediately within 10, 20, 30 million years, you get all these mammal lines that are starting to fill niches immediately. And so when you open up niches like that, you get explosive evolution. You get an explosive diversification. And so this is a phylogeny also. It looks a lot different than any of the phylogenies I've shown you to this point, because this is what they call an abundance phylogeny. And this is out of that textbook that I suggested you guys could get. And you can see these are some of the major vertebrates on top here. And this gives you number of species. And over here are time periods, right? So you go from when the planet was like 4 billion years old up to today, okay? And so what you can see is vertebrates, at least the ancestral vertebrates, are pretty old, right? About 500 million years. So first chordates arose. And vertebrates probably arose soon after that. And they were all what are called jawless vertebrates for a while, okay? And so this group here, Aganatha, this is going to be our most ancestral vertebrate group. And they are not diverse today. So you see how thin their line is? There's only a couple species. They used to be really diverse. But the organisms that outcompeted them up in this area was when the jaw formed. When the movable jaw formed, then those organisms started eating all the agnathids, right? Because that's when predation kind of arose. And this group here, chondrichthyes, are sharks and rays. And they're an incredibly fantastic group. What's really cool about them is if you look here, when they arose and started eating all of the jawless fish, they diversified and have stayed diversified pretty much during their entire evolution. And the reason for that, it took a little dip right here because there were a bunch of extinctions because of loss of prey with changes in ocean ecology, but also because at this point there were a lot of marine reptiles that were eating them, big, huge marine dinosaurs that were eating them. But still, you can see that they've been diverse almost uniformly throughout their entire evolutionary history. And the reason for that is because when evolution created sharks, it was almost a perfect aberration of match niching, if you will. They fit right into their niche immediately. They've always been apex predators. The only thing that sharks have done, the big ones, is shrink in size, right? Because their prey has shrunk in size. Would you go away? Say gradualism, punctuated equilibrium, make sense?